This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 73 for March 12th, 2010. Hey everybody, it's Vincent Racaniello, and this is TWiV. If you want to learn about viruses and how they cause diseases, this is the place to be. And joining me today, as usual, the reliable Dickson, Despommier.
Hello, Vince.
How's it going?
It's going very well.
You have the black shirts today. Are you looking for the Steve Jobs look?
Well, now that the iPad is coming out, I think I better wear the uniform.
And coming live from Florida is Rich Condit. Hey, Rich.
Hello, Vince. How are you today?
I'm well. And I understand it's raining where you are as well.
Yeah, well, not right at the moment, but it has been off and on the past couple of days, and it's overcast. I don't think it's done yet.
It's raining up here. You know, we always start out TWiV talking about the weather.
We do.
Why do you think that is?
Well, I have an explanation for that because I think we've emphasized climate change and weather events so often on our shows that it pays to start to pay attention to it on
I like that excuse.
daily basis.
Not just that we don't know how to have a conversation.
No, we can start with how was your breakfast this morning? You know, that sort of thing. But
the weather is far more interesting. Well, especially since
weather's a big deal, you know.
it's a giant deal.
Yeah.
You
betcha.
It drives people's lives, right?
It drives them crazy sometimes.
Mark Twain said, you know, everybody's always talking about the weather. Nobody ever does anything about
This
it.
is all true.
It's true. I don't do anything about it. I just talk. Well, today, of course, as usual, we have great virus stories. We have 4 of them. And Alan Dove, by the way, is AWOL. I sent him an email. Maybe he'll join us shortly.
He's probably out tapping the maple trees in his neighborhood right now.
Or maybe he forgot about us.
Nah.
I don't think that could happen.
If it's Friday afternoon, it's time for TWiV. So
Yeah.
That's
we're
true.
gonna do it. But we have 4 stories, and one of them is, again, a West Nile story
I'll
just
be
for
darned.
Dickson to
I'll
keep
be
you
darned.
happy. Because I know you fall asleep when you don't have West Nile stories.
No, I spend the most of the time asking questions out of extreme ignorance.
No. Yeah, out of extreme knowledge.
But not about viruses, so I don't pretend to know about
The first article was published very recently in Nature. And
Not a bad little publication.
not a bad publication at all. It's a fascinating article. It's called HIV-1 Infects Multipotent Progenitor Cells, Causing Cell Death and Establishing Latent Cellular Reservoirs. This is a big deal, I understand. Now, that's a complicated title.
It is.
Let's give a little background. As you know, we can control AIDS, the disease that results from HIV, by treating with antivirals. You can give people cocktails of 3 different antiviral compounds and reduce the amount of virus in them to under detectable levels.
Right.
And they can live complete long lives as a result.
Right.
But they still have virus in them. They have latent genomes.
So Magic Johnson still has
Magic
HIV.
Johnson still has HIV
Because
DNA.
he looks great on television. I mean, he's put on a little weight, but he's happy. He's ebullient. He's got his own self.
No, these antivirals work great. But if you stop taking them,
right,
what would happen?
I guess he would revert back into the
because he has
syndrome.
He's got a little bit of virus. In fact, now there's some more sensitive methods. You've got about 1 to 5 copies per milliliter of blood. I think that's the number.
Wow.
Let me look it up here. Just
And
to be sure.
that experiment has been done, giving people drug holidays
That's right.
to see if
Gotcha.
to see if it comes back, and it does.
It does
indeed.
come back, yeah. So these people on triple therapy, which is also called HART, highly active antiretroviral therapy, reduces viremia to 1 to 5 copies per milliliter of blood. So yeah, if you stop taking the antivirals, that comes back with a vengeance.
Yep.
So
That's as opposed to many, many thousands of copies per mil of blood for an active infection.
You bet.
absolutely.
Right.
Yep.
So the question has always been, what is making that virus? So there are cells that have genomes in them that produce the virus. They're latently infected, we call it. And some of those are CD4 cells, but there are also other reservoirs, and people have been trying to figure out what they are.
Are these the precursors for CD4 cells?
So these multipotent progenitor cells or hematopoietic progenitor cells, they're the precursors in the bone marrow of all the blood cells,
Right.
including the CD4 cells.
Right.
And this paper basically shows that those bone marrow cells can be latently infected.
You'd call those stem cells, I presume.
Well, actually, that's a good question because I was just looking over the discussion to this, and they waffle on that little bit of semantics.
Oh, whether it's a stem cell or not?
Yeah. They say, and I was confused about this. They say, are finding that HIV infects HPCs, hematopoietic progenitor cells, with an immature phenotype has clear ramifications for HIV disease, blah, blah, blah. Although further studies are needed to show that CD34 positive stem cells, which is stem cells, are infected.
Right.
Our detection of HIV genomes in HPCs, isolated from people, blah, blah, blah. So they're making a distinction between CD34 positive stem cells and HPCs. And I'll bet, I don't know, but I'll bet there's a lot of controversy over what's really a stem cell and what's not.
Right.
So HPCs are probably really, really close, and some people think and some people think they are stem cells, and some are not so sure, something like that. But for our purposes, close enough.
By the way, there's a connection here between a lot of things that we've been saying in the past and this particular article, in the sense that David Baltimore and Irving Weissman are fishing buddies, and Irving Weissman has isolated the 7 minimal number of cells from a mouse bone marrow that's needed to reconstitute the entire hematopoietic system of an animal.
That's right.
But he can't get it down below 7, so he doesn't know which of the 7 is actually the progenitor cell for all the other stem cell types that would go out and then. So this is
my guess is this is the same kind of conversation.
Yeah, exactly.
And David, of course, discovered reverse transcriptase.
See, that's
what's responsible for putting the virus in these cells.
So this is the Alan Dove part of the historical approach to this subject.
Well, they call these in this paper hematopoietic progenitor cells.
Yeah,
and they call them multipotent, which means that they don't give rise to all the blood cells, just some of them.
Sure.
So that's a good distinction, Rich, which I didn't appreciate.
But by the way, what makes us think that there's only a single cell that can give rise to all of it? What if there are 2 that give rise to just the T and the B?
Well, it has to start with one cell, doesn't it?
Not necessarily.
No?
It starts with an egg, that's true. I guess maybe that's where the thoughts come from.
I don't know. I mean,
that's a good question. I don't know.
I am certainly not an expert on this. There could be a distinction. But getting back to this paper, which they basically they purify these cells from human bone marrow, and they say that previously it was very hard to do this, and we have new cell culture techniques and flow cytometry, ways to purify single cells depending on what kind of protein they have on their surface using antibodies. Very interesting technology. And so they show if you purify these cells, I mean, these are CD34 positive cells.
Right.
And CD34 being a marker for stem cell-ness, right?
Yeah.
Right.
They can be infected with HIV in culture. They can be killed. They can be latently infected, which means the genome integrates. And when the genome's integrated, if you treat them with compounds that make them differentiate, the virus starts to replicate and be produced.
So at what level does the triple therapy work? Why doesn't it cure these cells also?
Because the triple therapy works against reverse transcriptase, other viral proteins.
And if it's not doing anything, it doesn't get killed.
Exactly. It's latent. It's just the DNA in the cell.
Okay. But if you keep the drug pressure on, doesn't it become non-latent at some point?
No, these are long-lived.
It just sits around.
Yeah, they're long-lived cells. That's the important thing about the paper.
Got it.
They live a long time.
Okay.
So you can have drug there, and the DNA just sits there and laughs at the drug because the drug isn't doing anything.
Got it.
And that's why we want to know where these integrated genomes are.
Sure.
So they actually, in this paper, also take bone marrow from patients that have been treated with drugs for years and don't have any detectable virus, and they still find latent genomes in this CD34 positive population.
So where are these 1 to 2 viral particles
in the
per
blood
mil coming from in the pressure of the drug?
Well, they're from cells that can start to make virus.
Yeah.
And of course, once the drug is found,
Then
is
it hits
encountered, it's nullified. So that's why it's probably so low.
So is there a way to turn on a latent virus?
That's the question. And that's why they want to find out where it is.
Completely cure the thing, right?
So maybe it's in multiple places. This is probably not the only place. It's also in CD4 cells, maybe somewhere else. But yeah, that's the key. If you can find out how to get it replicating, it will kill all these cells. Because as soon as you induce the replication, it will kill the cell.
Right.
And then with having antiviral around would presumably wipe it out.
Right.
So you might cure the infection that way.
I'd missed that idea. That's pretty interesting.
So people have been trying to do this with various interleukins for years to try and get stimulate T cell maturation. And the idea is that then you get virus replication. So maybe you could do the same in the bone marrow. Now, of course, if it's a big fraction of your bone marrow, you don't want to kill it off.
No.
Although you do, I guess you could do a transplant of some kind.
Yeah.
Well, that's an article we discussed some time ago, right?
Yeah,
where you
that's
can
true.
actually cure people with a transplant.
Yep.
Yeah.
So HIV downregulates interleukin 12. This I know from the toxoplasma story that used to be true. That isn't any longer because of the triple therapy. But so what if you gave interleukin 12? Would that?
I don't think it would affect the integrated genomes. That's the problem.
It wouldn't
No, they're
cause
still
the
there.
virus to become non-latent at this point. That's too bad.
See, in the last sentence,
So what's the purpose of the virus downregulating IL-12 then?
I don't
Does
know.
that serve any biological role for HIV infection?
It must. Viruses
Correct.
don't do things that are irrelevant.
Understood that.
But I don't know what it does.
So if you could restore IL-12, what does that do to
And
this
if
situation?
Alan were here, he could Google it while we're talking. But he's not, and I can't do I
Right.
can't
I
do
mean,
two
yeah.
things at once.
I'm just thinking about Magic Johnson once his 3 therapies stop working because of the mutation rate of
Well,
this
then he
virus.
switches-
It's going to
no,
overcome
but then
And he
this.
switches to 3 other drugs.
Yeah. So we have over 20, so you can make a lot of
Okay,
combinations
fine,
of
that's
3.
fine. I was worried
Yeah.
about him.
No, that's fine. I mean, the thing is, though, you don't want to have people on drugs forever.
Of course not.
So you would like to know how to get rid of the latent virus. It's so low, right now. If we could get rid of these, if it's a percentage of your bone marrow, you could induce them to replicate and
Yeah.
clear it. That
People
would be
on this
fine.
therapy can't transmit the virus. Is that true?
I don't know the answer. It's very low, though. 1 to 5 copies per mil.
Yeah, I don't know the answer to that either.
It depends. If you took their blood and injected it into you, you might be able to transmit.
No, I
it.
meant by the other common way.
Sexual transmission.
Yeah.
I don't know. It's a good question that I don't know the answer. But this is a big deal in the field because
Apparently
first
so.
time it's been
Yeah,
found.
Yep.
And apparently there are new techniques. And this paper, by the way, is full of very tiny graphs.
That's
Yeah.
right.
Every figure has a dozen
Can you
different
click
graphs.
on them and get the bigger version?
They're still tiny.
And we haven't explained most of it. It would take us several shows to do that. This would be a journal club that would go on for hours. But it's high state-of-the-art flow cytometry, being able to purify individual cells
Wow.
based on what's on their surface. This
Wow.
is done in a very expensive instrument with lasers that can detect 4 or 5 different colors, I think they're up to.
That's fantastic.
And that has revolutionized this whole area.
Let's hear it for Marty Chalfie.
This is not Marty Chalfie. This
No,
is
but they wouldn't see the colors if it wasn't for him.
The colors are
Different colors?
are not green, are not fluorescent
They're not
proteins.
based
They're
on that.
dyes. They're
I see.
small dyes.
Okay.
Rhodamine and
Okay,
fluorescein,
okay.
that sort of thing.
Okay.
But really nice stuff, right?
That's
I remember the first time I saw flow cytometry described to me, and I was just astonished.
I totally
with one
agree
of
with you, Rich.
that you could, this machine actually inspects one cell at a time.
Yeah. And pushes it
Decides
over. Yeah.
How
that's right.
many of which colors it
Yeah.
has on it, and then it can put that cell in a tube that you designate. Oh, I want this tube to
That's
have
right.
blue and green on it, and
Yeah,
this
yeah.
tube over here to have something else. Give me a break.
It's very cool. Yeah,
That's
the
right.
cells get passed through one at a time
Yeah.
through a channel, and then depending on what color they are, they get sorted into different pots.
I
It's
tell
just
ya.
fabulous.
It's the old answer to the question, tell me what you want.
Yeah,
I'll give it to you.
I'll give it
but
to
you
you.
better be sure of what you want.
yeah. You always have to be sure.
That's right.
Now, here's another cool paper. This was Alan's pick, and unfortunately, he's not here to do it. But it's a big DNA virus, so Rich will love it. All right.
Yeah.
The
Bring
latent
it on.
The latent human herpes virus 6 genome integrates into telomeres of human chromosomes.
Which are repeats, tandem repeats,
Yeah.
right?
Now, this is very cool. This is virus HHV6, human herpes virus 6. Rich, you know all about HHV6?
I don't know all about it. I know some about
Can
it.
you
I
give
was
us
wondering if we ought to put. I'd like to put this in the context of a short synopsis of the herpes viruses in general.
Sure.
Right.
Okay, so
That's a show
the
all to yourself.
herpes viruses are double-stranded DNA-containing viruses that replicate in the nucleus. They're pretty big. We're talking what? 90 to 100 to 150 genes or something like that. They have an icosahedral capsid and an envelope around that, and a very interesting space between the capsid and the envelope that the herpes virologists call the tegument, that has all kinds of goodies in it that are used immediately when the virus infects the cell.
Right.
There are herpes viruses of all sorts of creatures throughout nature, and they pretty much, to a large extent, stay with their normal host. They don't cross species very frequently. There are some notable exceptions to that. In humans, there are 8 herpes virus types that have different nomenclatures. You hear them called HHV1 through HHV8, where HHV is human herpes virus. The more common names are HHV1 is herpes simplex 1, that causes cold sores. HHV2 is herpes simplex 2, that causes genital sores. Right. HHV3 is varicella zoster, that causes chicken pox, or and later
shingles.
On shingles.
That's right.
Yeah.
HHV4 is Epstein-Barr virus, that causes mononucleosis, Burkitt's lymphoma, nasopharyngeal carcinoma. HHV6, which we'll talk about, causes a skin rash called roseola, as does HHV7, as I understand it. And HHV8 is Kaposi's sarcoma-associated herpes virus. That's the herpes virus that shows up in these lesions that are in immunocompromised people, in particular a diagnostic initially of AIDS.
And all these viruses hang out in the nervous system if they're not...
not all in the nervous system, but they all have in common. Besides, I mean, a kindergartner could recognize one of these in an electron microscope. They all
big.
look very much the same.
Right.
Okay. What they all have in common is that they establish latency.
Got it.
Okay.
Yep.
So they will infect and undergo an acute infection where you get productive virus replication, and then establish latency where the genome is harbored in the nucleus of some sort of cell in a quiescent fashion, doing little, if anything. And then at various times in the future, under various circumstances, they can be reactivated. Usually the circumstances are stress, and they'll start to replicate again, and you can get symptoms again. Now, a lot of these can be asymptomatic forever, like herpes simplex virus. Almost everybody's infected with herpes simplex virus, but only about 15% of people get the symptoms, which are cold sores.
Right.
On the other hand, before the days of vaccination, almost everybody got symptomatic chicken pox. That goes latent and can emerge later, as you pointed out, as shingles. It's only a small percentage of the population that shows up with shingles. Some of these establish latency in nervous tissue. Right. That includes herpes simplex types 1 and 2, the cold sores and genital sores, and varicella zoster virus, the chicken pox. That
does this cause Bell's palsy also?
That rings a bell.
Excuse me.
That was. I didn't even know I was doing that. That was Alan.
That's great.
We'll give you a clapper for that.
To look it up. I can't multitask that.
Okay.
And some of them establish latency latency in lymphoid tissue, B cells, T cells, etc. That includes EBV, cytomegalovirus, and these and the rest of them, HHV 6, 7, and 8. I believe all of those. So HHV 6 is one of these. Oh, and what's known about latency in all of these other viruses, in the capsid, the genomes are linear double-stranded molecules. During lytic replication, there's been a lot of discussion about this, but people think during lytic replication or active replication, the genomes are replicated as linear molecules. But when they establish latency, the genomes circularize, and they are not connected with the chromosome. They're sitting in the nucleus as what we call episomes. That is circular molecules that are not covalently hooked up with the host chromosome.
Very interesting.
So HHV6 is one of these viruses. It's a fairly recent discovery. It's, I believe, causes this skin rash called roseola. I think there's a fairly high infection rate in the population.
90%.
Okay.
By age 2.
And what this paper deals with is the mechanism of latency. And what's really unusual about this and really cool is that they find that this virus, when it goes latent, actually integrates its linear genome into the telomeres of chromosomes.
Amazing. That is amazing.
And what I like best about it is that it shows that it can be transmitted vertically. That is from parents to kids.
So it's in the germline then.
Right.
Yeah.
And so the way they do that is that they have families that they've analyzed, 4 of them, where they have fathers, mothers, and children, and they've determined by a technique called fluorescence in situ hybridization, FISH, which is also a cool technique. That means that you spread out chromosomes in a cell, and you hybridize to that a piece of DNA that homes to its homologous location on the chromosome, and that has attached to it a fluorescent probe, and that'll light up in a fluorescent microscope so you can see the chromosome and this color really bright where this thing lights up. So you can map genes that way. And they show that in these families, you can, for example, here, family number 1, father has this thing in a location on the 18th chromosome called 18q23. The mother did not have HHV6 integrated, and there are 3 children here, all of whom have HHV6 on the 18th chromosome in the same position as dad.
Amazing.
Okay, and let's see, they've got another one, mother, child on 17p, another family, mother, father's negative, child's positive on 22q, same as the mom. Very cool.
So the implication is that that was inherited, right?
Yep.
But
absolutely.
none of them in the same spot, so there's a randomness to the integration site.
This is as far as they can do it. Didn't they sequence some integration sites and show?
Yes, they did. That's one of the things that told them they were actually in telomeres.
Got it.
Okay. And they look located to telomeres, but they can see the telomeric repeat sequences adjacent to the integration sites, if I understood it correctly.
Okay.
Right. When this DNA is integrated into the chromosome, my understanding is it still can produce infectious viruses, right?
Yeah, because these families were chosen because the kids all really have high viral loads, I think. So, yes, they can make infectious virus. But how you get from the integrated to a replicating copy,
that's what I wanted to know.
I don't know.
Onion skin?
Yeah, maybe something like that. The chromosome has on its left and right arms direct repeats.
Right.
Okay, so there are opportunities there for regenerating the repeats if you can't quite replicate them out perfectly. This onion skin replication that Vince refers to is like if you have an origin of replication in the middle of this thing, you could replicate that out independently of the rest of the chromosome. So it's hard to describe, but you can imagine this bit of DNA replicating out and then a bit of DNA replicating out of that. So you get this layers of replication out of the original site. Right. You might imagine it looks like an onion skin.
Right.
Yeah, I'm looking for a picture for Dick here because he's making faces here.
Yeah, I was just curious. I hadn't heard that.
Just show him an onion.
I know.
An onion has many layers. So you basically, when this replicates, you get many copies of DNA. So here's the interesting question. I noticed a part of this paper where they said the results suggest that the virus integrates into the host genome via homologous recombination with telomeres. The perfect TTAGGG repeats encoded in the right and left direct repeats of the viral genome mirror the telomeric repeats. So, Rich, does that mean that the virus has a sequence which is the same as the telomere repeat, and that's why it's integrated into this?
That's what it sounds like to me, yeah.
That's amazing.
So it's a homologous recombination. So that implies a little teleology, like it's designed to do this.
But so that would be why
telomeres.
the virus is going into the telomere. Do you think there could be any other reason? The telomere is maintained, but it gets shorter and shorter as you age, right?
Right.
Or with each cell replication.
Would there be another reason why this DNA would like to go in a telomere? I don't know the answer, but
no, not that I can think of. Man, now I don't know what telomeres are not. Well, no, wait a minute. They have something in here about transcriptional activity of telomeres.
Yeah, they say that there's an RNA.
Telomerase.
That there's an RNA produced called TERA, which regulates telomerase and many important telomere functions.
Yeah.
And they say it's possible that integration of the virus could alter the expression of this RNA, leading to dysfunction of telomerase. So maybe this would make the cells immortal.
Exactly.
And that the virus would like to do that, right?
I actually was going to ask that question, but you beat me to it, Vince.
Because many viruses make cells immortal so that they can be produced forever, right?
That would be a great idea.
But man, if this is in the germline, that would that would imply that. I mean, that would imply it's in every cell.
Yeah. So, what is the actual cell of this particular article? Which cells did they actually find them in where they did the study?
Well, that's a good question.
Let's see. We can look in the methods here.
They're taking peripheral blood T cells.
T cells.
T cells.
Yeah. So the good question is, what other cells?
Right.
Because if it's being passed in the germline, then theoretically you can have it in every cell. If you immortalize every cell, you're in big trouble.
That's so.
I don't know of any malignancies associated with this, but
no.
Perhaps the integration prolongs the life of
Epstein-Barr virus,
cells.
though.
Yeah.
They do.
Burkitt lymphoma.
They do say that. Now, I didn't read the text on this, but in the table they describe some of the people as having CNS dysfunction, hypersomnia, ataxia. Some mostly CNS looks like disorders of one sort or another. Now, whether that's due to the virus or not, I don't know.
Yeah, they say subtelomeric rearrangement has been associated with mental retardation. The other thing they wonder is whether the kids who have this virus from birth, because they're inheriting the DNA, whether they have any autoimmune problems as a consequence.
Right,
because it's going to look like self.
Yeah, exactly.
Exactly.
So I wondered, if this is in the germline, the first thing I thought of was, when did it go in the human germline?
Now we're sequencing Neanderthals again.
Exactly. So to do that, you have to get other species related to humans where this virus is integrated and try and build a tree. So
absolutely.
I emailed this morning Peter Medvexy. Okay. And I just got a response. So let's take a look at this. I said, can you give us some information about if other species have it? I believe there are similarities between retroviruses and HHV6 as they both integrate, but I believe HHV6 integration is different and is an ongoing process. The virus is present in only one of the 46 somatic chromosomes, so the offspring has 50% chance of inheriting the virus. I believe that HHV6 can cross the placenta. Then the fetus may acquire the integrated viral genome in the pre-gonad cell. Then the integrated virus is passed to the next generation. HHV6-like viruses are present in primates, but nobody studies them. Okay.
Well, it's time then.
But he's saying this is probably an ongoing event. It didn't go in there 50 million years ago and then was passed on forever. But it would still be interesting to see in these primate viruses what they look like sequence-wise in comparison, if you can get some idea of how long this has been going on.
Right.
So thanks for that answer, Peter. Just in time for TWiV.
Hey.
It was great. Excellent. Okay.
So that's this is a very nice paper. That really
beautiful.
Is a unique perspective on latency and herpes viruses. It's going to. I wonder what sort of impact this will have on other people studying latency with other herpes viruses. Yeah. It's pretty well established in most of the other cases that it's episomal, but this is very interesting.
Okay. A great paper. I love it. Now, before we move on to our last story, I'd like to acknowledge the support of TWiV by Data Robotics, the makers of Drobo. the wonderful, how should I put it?
It's an extra brain for your computer.
An extra brain, a storage solution for your computer, whether it be a Mac or a Windows or a Linux computer. It's a very flexible and safe backup solution, and you can also work on it. Drobo comes in different sizes. Go to drobo.com/TWiV to have a look at what kind of backup solutions they do make. It's a small box where you can insert any number of drives you wish, and the data is then redundantly stored on these drives so that if one drive fails, you can actually pull out the drive, replace it with a new one, and your data will be restored. I have 2 Drobos at home. I have one here in the lab, which Dickson is looking at this moment.
I am, actually. I just moved the chair out of the way, and I opened it last week just to make sure
and
that it had something inside.
There's one waiting for you, Dickson.
This is
to
true.
Put on. So why do I have 3? Well, here at the lab, I store all my data, all the PhD theses of my students, all my lecture notes, all my recorded lectures, and I'm very happy that it's on that because it's safe. Now, in the past, I used to have an external drive backup, but I was never sure that it would be safe. And I've always lost external drives to crashes and lost data. So I'm really happy that it's secure, and I recommend anyone that has a research lab which generates data, and by definition you will be generating data, to use a Drobo to back it up.
Right.
And if you go to drobo.com/TWiV and use the promo code Vincent, you can get $100 off the purchase. However, Data Robotics have really been generous, and they're giving us 3 units to give away.
Superb.
And we've given away the first one already, which we announced last week.
Yep.
And the second contest is ongoing. And you should go to twiv.tv/Drobo contest for the rules. But essentially, write an essay, no more than 500 words, to answer the following question. Would it be possible to genetically engineer influenza virus so it could replicate in bacteria? Why or why not? So we've gotten a couple of entries already. You have 3 more weeks to enter this contest, and then the TWiV team will grade them.
The TWiV team.
Because we are academics after all.
We are. We're good at this.
And we know how to grade. And
get out those red pencils.
The highest score will win the Drobo S. And then we'll have a third giveaway where we'll do something different so that other people can compete in this as well.
We have a random selection afterwards, perhaps.
So that's a lot of fun. Here come the helicopters.
Yep.
That's a lot of fun to answer that question, and we've gotten a couple of interesting submissions already. So sharpen your pencils, open your laptops, and answer that question.
Sharpen your wits.
Sharpen your wits, too. And we thank Drobo, Data Robotics, for their support of TWiV.
Right.
Story number 3. Aha, this is for you, Dickson.
Thank you.
Do you prefer Dickson or Dick?
It doesn't matter. As long as it's not said in a menacing way, I'm fine with either one of those things.
Menacing way. I'm not menacing whatsoever. No. Let's see. Let's get up this paper here, which is now it's gone. It's dispersed. Population genetic data suggest a role for mosquito-mediated dispersal of West Nile virus across the western United States. This is right up Dickson's alley because he wrote the book. What is it called? West Nile Story.
Right.
Now, West Nile came to the U.S. in what year?
1999.
Appeared here in New York City.
It did.
Right?
Yep.
And then since then, it's spread across the whole country.
It did. Very fast.
How long did it take?
It took about 4 years. According
to this abstract, it said the westward movement is thought to have been mediated by random dispersive movements of resident birds. Are you shaking your head? You don't?
When this first happened, I'm so happy to see this kind of study. I made a speculation, which I submitted to Time magazine.
Really? Did they publish it?
They did.
Was it probably not peer-reviewed, though?
No, hardly peer-reviewed, but it did elicit a phone call from a former friend of mine who had dropped out of sight for a long time and reemerged as a result of seeing this letter to the editor. And what I suggested was that the east-west highways of America, particularly Route 80, have these big drainage ditches in the middle, right? So this is a mosquito that loves pollution.
Culex tarsalis.
Culex tarsalis in the west coast, but Culex pipiens in the east coast, and some interbreeding mosquito in between there. But it's always Culex. The Culex mosquito loves polluted water. So somehow you have to get the vector and the virus and their hosts together And their biggest host is birds, and one of the biggest bird hosts is crows. So crows, you travel out west sometimes, Vince, maybe western Pennsylvania, western New Jersey, western. Everywhere you go, you're driving along and you see crows. They're all along the side of the road, and they're picking stuff up from the grass. What do you think they're picking up?
Mosquitoes, bugs,
No.
carrion.
Carrion. That's exactly what they're
Do
doing.
you hear the joke about the two crows that...
Those were vultures. No. What they're picking up are the dead bugs that bounce off your windshield.
I see.
No, you're rich.
I thought you said they were carrion.
That is carrion.
It's just small carrion.
Bug carrion. And you've seen the results of them on your windshield.
I said bugs, didn't I?
Right? Remember the song, Sometimes You're the Windshield and Sometimes You're the Bug? Okay. So in this case, these debris are created by the cars and the trucks on the Trans-America Highway system. And so you've got the perfect combination of draft from the cars, dragging populations of mosquitoes from one place to the next, laden with viruses that then establish new colonies in the drainage ditches, waiting for the crows to come to feed on carrion and to give them the next round of virus. And that's how this thing went from the east to the west. That's why I said in this short letter, son of a gun. Look what we're reading about now.
So they say here that the previous idea was the bird, but they thought that it could be mosquitoes.
Yes.
And they're focusing on Culex tarsalis.
Right.
They say it's capable of moving relatively long distances.
It has a long flight range.
So
the next sentence is the sentence that really gets me in this paper.
A series of mark, release, recapture studies conducted in populations in California showed that females can travel several kilometers per night for consecutive nights while hunting and seeking oviposition sites. Cool. What's an oviposition where they can lay their eggs?
The water. Yeah.
And
so females can travel several kilometers per night. That's incredible.
They can fly far.
And they say this level of dispersal.
Dickson, how do you do a mark, release, recapture stuff? How do you mark a mosquito?
Oh, you put a little colored dot on them.
Oh, really?
Paint. Yeah. No, they've done this with bees and ants and termites and mosquitoes.
You must have to mark a lot of mosquitoes to recapture them.
You do, but it's a fluorescent dye, so you can actually capture in a mosquito trap millions of mosquitoes, and the one that's fluorescent you can pick out of the crowd just like your cell sorter. All right?
So they say, Dickson, that this level of dispersal is not much different from bird movement, which ranges between 0 and 14 kilometers.
Right.
I guess per day.
Right.
So that's why they say maybe the mosquitoes spread it.
Well, I would have added another reason there, too, because of the draft of cars are just moving these things along.
So what they do in this paper is, this is way out of my specialty, but I understand the principle. They look at microsatellite loci in the DNA of these mosquitoes. That is, it's all one species, but there's slight differences.
Correct.
That they can track across the country. And they actually did it. They actually did it in the West.
Yep.
And they identified the movement of genes, basically, in the mosquito populations.
Yep.
It's amazing.
You can also introduce a crossover in the lab that has no bearing whatsoever on reproduction.
Oh, and then release them?
And then release them and watch for the crossover showing up.
So they say we found that the observed pattern of genetic movement mirrors the invasion pattern for the virus across the western U.S. between 2002 and 2004.
Right.
They basically say that. So this doesn't prove it, but it could have accounted for the movement of the virus.
That's right.
Not just the birds.
Exactly. But remember what they also did. They collected lots of mosquitoes in various places, and they looked for the virus. So what I would like to see...
They didn't do that here, though. No, some other studies, right?
Yeah, the CDC was conducting those studies, and I haven't seen those released to the public yet to show the actual movement of the virus from east to west in mosquito populations. The only news we ever got was human infection, basically.
Now, Dick, look at this map here on the page.
Yeah, I'm seeing it.
Page 9 of the article.
Yeah, I've been looking at that. It's just fascinating.
Distribution of West Nile incidents in humans and reports of West Nile activity, which is green, by county and year.
Right.
So 2001, 2, 3, and 4. Dots represent human incidence rates per million. You can see the virus spreading westward.
That's right, like a wave.
And then the black vertical line is the eastern boundary of the tarsalis movement.
Yes.
So it doesn't go any farther east than that.
No, it jumped from Culex pipiens to Culex tarsalis, which is we would call them not sibling species, although they do look very, 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 very similar, but you'd call those ecological niche occupiers of the same niche in different geographic regions. So they both love polluted water. But
so how did the virus get across this barrier then? Went from
cars.
No, but the Culex don't go across the barrier, and the pipiens don't go across the barrier. Tarsalis, I mean.
So you get out. Wait a minute. You want me to describe this to you?
No. So basically, the mosquitoes bring it to some birds, and then they reinfect other species of mosquitoes and carry it farther westward.
That's or you open your car door, and because you've had too many beers, and you pull along the side of the road to
take a leak.
Miturate, well, I'll use the polite term here, and your car door is open, and the mosquitoes are flying around like crazy. They didn't get into your car. You drive off, you cross the Mississippi River, you open your car door, voila, you've got some infected mosquitoes to give to the birds, could then give them to the tarsalis mosquitoes.
Then this curved line is the observed barrier to gene flow among the mosquitoes. You can see in 2003, it stopped the virus from, that corresponded to where the virus stopped. And then eventually it got across that. As well, because I presume other mechanisms come into play.
That's this one. I'm just
the cars.
So what would those barriers be? Are those like geographic?
Yeah.
Or
the Mississippi River basically is what you're looking at there.
No, this is not the Mississippi.
No, not down over there. I meant
the straight line may be the Mississippi.
In 2002.
Well, what is the barrier? That's a good question.
Well, desert. We've got a desert.
Subspecies. Deserts. Oh, yeah, that's
Don't forget about the desert.
And the Rockies go through there, too.
They seem to.
Well, they're further west.
That's true. And by the way, as you drive west, the need for a drainage ditch disappears. So when you get into the state of like Wyoming or into Nevada, the road is there, of course, but there's no drainage ditch anymore. So there's no way for the water to accumulate, even in temporary basis.
It's geographic.
Right? It's geographic.
So what they say, gene flow in tarsalis appears to be extensive, potentially mediated by movement of mosquitoes among neighboring populations and hindered in geographically limited parts of its range.
Right. So I think we do the rest.
Okay.
And it's slower this way, of course. But, you can have mosquito-induced malaria at an airport. You know how that happens, right? The airplane stops in Ghana on their way back to the United States. They're there for 2 hours. Sure. The doors are open. In go the mosquitoes. The next thing you know, they fly out of the airplane when it lands at, let's say, LaGuardia. And the next thing you know, you have...
Has that happened?
Yeah, many, 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 many times.
Oh, I always wondered about that.
Many times.
I didn't know that.
Mosquito-induced...
Any other diseases besides malaria?
Dengue.
Really? So at the airport or just peripherally around the airport?
No, due to air traffic. They said one of the funniest, I guess, slides I've ever seen when I first saw it, not anymore, is a Boeing 707. And the guy giving the talk said this is the world's most efficient vector for diseases.
Yes, but even before there was...
Not because they travel with us, but because they take the vectors with them.
Before there were airplanes, diseases still spread.
No question about it. Oh, yeah, because we refuse to take no for an answer. We go everywhere.
We travel, but it just was a little bit slower. But
that's right. It was slower.
That's how Cortez conquered the Aztecs.
Exactly.
In quotes.
Exactly. So for many years, there were certain viruses were not present in the Western Hemisphere.
That's correct.
Until the conquistadors brought
them.
So
why is there so much controversy about what happened to Neanderthals?
What's the controversy?
The controversy was how did they suddenly disappear? They were disappeared concurrent with the appearance of humans. But there's no evidence for conflict between humans and Neanderthals.
Yes, disease, right?
Of course. It's the simplest explanation, and no anthropologist is using that as a way of saying what happened to them, but they should be.
But so that it implies that the two had to come in contact at some point, right?
They did. Of course, there's plenty of evidence for cohabitation of various spaces, various areas of Europe. But the interesting part of that is, and this is a sidelight, but think of how robust and rich our MHC complex is, genetically speaking, right? That's why we're so protected against all these infections that we're talking about.
Yes.
Right. Now, guess what? They've got 6 different sites where they've dug up Neanderthal bones. Mm-hmm. And in the skull, they can extract enough DNA to get a reading on the MHC complex. And it's incredibly restricted.
Yeah, sure. They're like an inbred population.
Correct. I mean,
exactly.
And their narrow range of ability to respond, coupled with our large range of ability to respond, is rich as comment. The conquistadores came to the New World and wiped out 45 million people. They didn't make enough bullets for that.
Even today, there are populations with more restricted MHC polymorphism,
Of course.
and they're more susceptible to certain diseases.
Well, every time they find a new primitive, I wouldn't call them primitive, but naturalistic population of people living in the Brazilian jungle, they stop and they get somebody who's been there a little while longer because they use them as the interpreters. They don't want to contaminate them with their own microbes.
So, Dick, I'm still looking at these maps on Figure 4. What you said, West Nile showed up in 1999. Is that right?
That's correct.
So 2001, this map's got all kinds of colors on it, indicating West Nile activity, green and orange and red. And in 2003, it's just really brilliant.
You're
in
right.
2004, it's more dispersed and sort of dying off. What does it look like now?
Even then, what you're really looking at is the pattern of drought. Every red dot that's a human case was acquired in a place in the United States that was suffering from an acute drought. And if you do go back to the National Weather Bureau, they have a drought watch map that they publish. You can go to the archives and you can take out that month, superimpose it over where West Nile popped up in people, and it's an exact match.
So there's sort of it's now basically endemic in the U.S. and pops
totally
up.
Endemic. It's in California. It'll never get out of the Central Valley because that's a drought-ridden zone every year, and they've got big cases. Texas has large numbers of cases every year also now. And everywhere else, if there's no drought, or if the temperature is cool below, let's say, 90 degrees, and it never exceeds 90 degrees, there isn't a single human case. If you look in the wildlife, it's all over the place.
And we talked about the influence of drought on transmission last week, if you recall.
I do recall that. So this is a follow-up article, folks, and I love the synergy that this literature allows.
Yeah, it's great.
And this
format, this kind of conversation allows us to be very flexible,
exactly,
and spontaneous.
Exactly.
All right, let's do a few emails, shall we?
Absolutely.
Chris from ASM. Remember Chris? He filmed us at ASM.
I do remember Chris.
He writes, I just received 4 pounds of fresh, flash frozen ground venison meat. I knew I wouldn't be able to say that. Say that fast. Fresh flash frozen.
Baby buggies, rubber bumpers.
And he wants to make venison chili.
Right.
The deer was killed in Virginia, which has one official report of chronic wasting disease.
Yeah, right.
Do you remember that? It's a prion disease.
Yes,
According to my hunting friend, an experienced butcher, which he uses to prepare his kill, can notice signs of chronic wasting disease in the meat muscle itself. Do you know if this is true? Also, since I do have these 4 pounds of lovely-looking venison, how would one go about getting the meat itself tested for CWD infection? Could a university lab do this, or is this a more specialized procedure? Oh, nothing is more specialized than university labs.
That's right.
Finally, if you are inclined, I'd be happy to send you all a pot of my venison chili in return for answering my questions.
So,
take it, Rich.
You know, one of the if you look under the diagnosis of chronic wasting disease or the symptoms, I mean, I don't know, I'm no butcher, I'm no veterinarian, but it's called chronic wasting disease because the animals are wasted. Right. And they show up emaciated.
That's right.
So it wouldn't surprise me a bit if an experienced butcher could see the disease, at least in fairly advanced stages, whether you'd see it in early stages, and whether the meat under those conditions would be dangerous, or I don't even know if it's dangerous at all.
Right.
Is another question. I'll pass on the chili.
If you hate it, you might pass on the chili. No, that was another.
So you don't like chili, or you don't want to take the risk?
I love chili.
So do I.
I'm not sure I want his,
It's interesting that this should come up at this moment because there's another related article in ProMed signaling the arising of another series of cases of BSE in cattle in Alberta. Now, this is rural Alberta. So I've often thought, is the rural cattle of Alberta exposed to cervid wasting disease, and is there a relationship between those two
So it's not
things?
because they were fed repurposed feed?
No. That's exactly right. So how different is this prion from the one that causes Jakob-Creutzfeldt syndrome?
It is not, well, the difference, and you only need one amino acid change, most likely,
That's
to
what I
make it
thought.
infect people.
So the hunters of North America are placing themselves at risk. Of course, you wouldn't shoot a damaged animal that looks wasting. And
Well, that's one of the guidelines. There's this Chronic Wasting Disease Alliance website that provides information basically for hunters.
Right. And
they say that, there's no evidence that it can spread to humans, and the risk is probably very, very small if it exists at all. And then they say hunters should take precautions when field dressing your meat, but this is very interesting. Concerns over CWD shouldn't stop you from enjoying the hunting season. So here's the recommendations, right? Sorry, go ahead, Rich.
I was, along with this email, was sent this news release about this deer from the Virginia Department of Game and Inland Fisheries, and I was surprised. Now, maybe this is just Virginia, but how rare it apparently is. Since 2002, nearly 5,000 samples have been collected in Virginia, and CWD has been detected in only one deer.
Yeah,
interesting.
2009.
So it's not, at least in Virginia, not a real high incidence.
So according to the website on the Chronic Wasting Alliance, you can contact the local agriculture division in the state, and they will test it for you. Right. I mean, it says whether or not they will test it depends on how busy they are, but that's the best way to do it, Chris.
I can't do it in my lab.
The National Wildlife Service might have a connection back to the National Diagnostic Labs also. There's a National Vet Lab in Ames, Iowa.
But
test it for you.
They say basically minimize handling of brain and spinal tissue when you dress it, bone out the meat, don't saw through bone, wear gloves.
Yeah.
Avoid consuming brain, spinal cord, eyes, spleen, tonsils, and lymph nodes.
By the way, the people that are most at risk on this one are taxidermists. Because they're mounting the heads of all these animals, right? They've had to pull out the brains and everything else in order to do it.
So go to the Virginia Department of Game and Inland Fisheries, Chris, and give them an email. And they will tell you what to do with it. There's information right here about how to get it tested.
Yeah.
Would I eat that venison? Sure.
You would.
Yeah, I don't. I'm assessing the risks. You have to balance
you're probably
things.
right.
I don't think it's.
I take
a
it
big
back.
risk.
I'll take some chili.
Would you eat it, Dick?
Send it to me.
How about you, Aaron? Yeah, he would.
Aaron, everybody here would.
All right. Our next email is from Nissin.
We're gonna die.
Nissin. Rich, is that how you pronounce it? Nissin?
Nissin.
You know this man, don't you?
I spend more time with Nissin than I do with my wife.
Well, don't.
We're going to cut that one out.
It's a different circumstance, okay?
This doesn't matter, Rich. I hope your wife doesn't listen to TWiV. My son sent me this article, and I thought it was fascinating. I don't know if it's a program or a FYI. Thanks for doing TWiV, Nissin, who's from Florida. I guess he's working in your group, right?
That's correct.
Thanks, Nissin.
So I've known this guy for 30 years.
This is lucky you. That's always nice to have that kind of personnel.
It's fantastic.
Look, tobacco.
So there, in this article, scientists grow cheap biodegradable solar using tobacco. They've engineered tobacco mosaic virus to build chromophores, which are the structures that will do photosynthesis. So they basically infect tobacco with the virus. The photophores are made, and then they extract them and spray them on a surface. And it's sort of like a photosynthetic coating.
Really?
Do you remember the viral batteries that we talked about last year? Same idea. These are rod-shaped viruses that align very nicely, and they can make photocells basically out of them.
How neat.
They aren't as efficient as silicon cells, but a lot more eco-friendly. They're biodegradable.
Well, that might be a problem, though.
Very cheap. So you have to harvest them from inside the plant, dissolve it, and then spray it over a substrate.
Right.
Now, Angela Belcher, who's from MIT and who has done the viral battery work, says this is not easy to do, and it's not clear that they've gotten it to work yet. I think this is a
work in
they've
progress.
grown the chromophores in the plant,
right?
And it's not clear to me if they have actually reconstituted photocells or not. But it's a brilliant idea, don't you think?
Yeah, it's really interesting. What wasn't clear to me is whether a single virus particle or as a chromophore, or whether they're talking about a crystalline array of virus particles that have this activity. Could you tease that out of this?
I couldn't. No, I tried to find an article on PubMed relating to this, and I didn't. There's no name here.
I would
right?
put this under the heading peaceful uses for tobacco. Do no harm.
They don't have to use tobacco, but the
It's
virus.
a big leaf.
It's a big leaf. The virus is a well-known
Sure.
virus, and we know how to handle it and
We do.
modify it genetically. So I suppose that's why they do it. I
That's
did
right.
try PubMeding this, but I didn't find anything.
My guess is that the actual structure of the virus is important in all this, because this is
Sure.
a long helical virus,
That's right.
a rigid helix, and
Yes.
that probably has that. From what I can get out of this, has properties that are relevant to its behaving
Yeah.
as
Yeah.
basically a
That's
photocell.
the case with the batteries. They used bacteriophages, which are rod-like in structure, the same thing, and they line up and make anodes and cathodes. So
And TMV crystallizes really easy, I think.
Yes, it does.
Was the first virus crystallized,
it
right?
was.
Nature's
And
nanotubes.
purify it, and it just falls right out of solution and crystals.
First virus discovered, first virus crystallized,
That's right.
first virus shown to contain RNA.
That's right.
First virus smoked.
Probably.
That's right.
It is.
Exactly.
Anyway, that's a great story. Thanks, Nissen. And keep listening to TWiV as long as you get your work done,
Exactly.
Yeah,
because Rich wouldn't like
Should
it.
have him on if he's in your lab, Rich. Invite him on next time.
we can do that.
Next email's from Ilya, who writes, Hello, TWiV hosts. In episode 66, you've been talking about retroviruses. I just encountered a really cool video on YouTube that can help TWiV listeners better understand the HIV replication cycle.
Cool.
And this is in Russian. I guess that's where Ilya is from.
Targeting HIV replication.
Okay, we don't.
But it's in English, though.
It's in English, and this is actually a well-known video. I've shown this to my class,
Oh, okay.
so I thought we would put the link out for everyone. It shows the whole replicative cycle in beautiful
It's very nicely done.
Nicely
detail.
graphed.
This is one of the better videos that I've seen. I think it's
Terrific.
originally produced by Beringer Ingelheim.
That's correct.
Oh, yeah.
They give their credit at the end. It's a very nice video
Yeah. Thanks for that, Ilya.
and very accurate.
Norman writes, now, too bad that Alan isn't here for this one. I suppose given the number of computer scientists who listen to TWiV, you may have gotten a number of responses to Alan's question about evolving computer viruses. So we said a few episodes, do computer viruses evolve? Nonetheless, since I did my university research on evolving neural networks, I thought I'd have a go at responding to the questions. So he wrote a blog post on his blog addressing this question, which is really nice. I read it. I tweeted it today. I posted it on Microbe World. And can computer viruses evolve? His basic idea is, the short version is that the real-world computer software is too fragile to be subjected to the harshness of evolution. So it's code. If you change the code, it won't work.
Right.
Yeah, that was the one conceptual thing out of this that really struck me that I really got is too fragile. It's a nice word to describe it.
Or
One little bug,
Yeah.
and the whole thing is down.
there's
See,
And
no
biological systems are a lot more
there's
malleable.
no redundancy.
Exactly.
There's no redundancy. That's right.
Yeah, we're more flexible. Absolutely.
Exactly right.
See, computer systems are binary. Life is not.
Nope.
Right? However, he continues, there are ways to represent systems in software that can be evolved, and it's often surprising, at least to those of us on the computer science side of the fence, how well evolution does work when given the right tools. So I direct everyone to his post, which is really nice, and I think it's great that we inspired someone to do a blog post and think about a question in addition,
You
right?
bet. You bet.
Thanks for that, Norman. We'll pass it on to Alan Dove. Ariel writes, hello, TWiVs. So Ariel is from Israel, and she says, some episode before, you talked about the emergence of mumps in Israel.
By the way, Ariel could be a male too, by the way.
Oh, I'm sorry. Well, at the end it says Ariel and ILA. So they could both be male, they could both be female.
Exactly.
It could be female and male, so I'm covering it all here.
Thank you.
You could let us know what the story is so we get it right.
Right.
So there's been yesterday, which is February 22nd. We're not that far behind. An ultra-Orthodox site published an article about the current spread. They say that dozens of yeshiva students were infected and hundreds are not vaccinated. The Ministry of Health decides to vaccinate all the students that were in contact with the infected students. So she sends a Hebrew article, an article in Hebrew in a bad translation. And so we'll
Really
pass it-
bad. I tried reading a translation. I could barely make sense out of it.
Google just came out with
It's a Google translation.
Yeah.
Well, they just came out with a new translating tool, so maybe they'll improve this.
Yeah, this is all Hebrew here. But then there are two links at the end. Let's see what these are. Is this Hebrew or English?
They're in English.
Okay.
Aha.
Israel is in the middle of a mumps outbreak. So we will put these links in for everyone to see. And perhaps your friend at CDC will have a look and give us some more insight. Thanks.
Right. These were it's the same sort of thing, crowded conditions, and a lot of these people unvaccinated. So it's a site where this stuff can really grow, though quite a few people who'd been vaccinated once or twice were also infected. So similar deal as we discussed before.
Here.
Thanks, Ariel. Prasad writes, "Hi, guys. Great podcast. Just stumbled onto this podcast and loving every minute of it. I am slowly getting up to date with the episodes during my commute. It keeps me informed about general virology literature and gives broader perspective. I'm currently a researcher at Duke NUS University in Singapore working on dengue."
Right.
Anyway, in this week's podcast, there was discussion about changing behavior of host by pathogens. So
I remember that one.
Remember, it was we're changing the aphid behavior.
Yep.
So we asked, are there any other examples?
Yeah,
so he said, how about rabies? I remember reading that rabies affects and somehow inhibits the fear center of infected animals and thus increases the likelihood that the animal can bite some other animal and pass on the virus. What do you think? Keep up the good work. Yeah, so when a raccoon is rabid, it has no fear of humans any longer.
Right, and attacks them.
That's right.
I hadn't thought of that. That's really great.
Yeah. That's a good point. Thank you.
By the way, speaking of that, I was just curious. The other day I was having a conversation with somebody, and since we know that the little brown bats are their natural host and don't suffer from rabies, and yet there's a lot of raccoons out there that seem to come up with rabies, how does the bat transmit it to the raccoons?
Well, maybe in a cave there's an aerosol. The raccoon goes in the cave.
Or?
Does the bat bite the raccoon?
I don't know, because the raccoon feeds on crustaceans along rivers. A lot of them do, at least. Not the ones around here, but the ones certainly in the wild. And bats are all over the place, and they may actually come in to try to take the food.
Could be.
I don't know the answer to that at all, but I know that they don't catch them and eat them, so I know that they're not transmitted by
I don't know.
by that.
Maybe somebody out there listening will know.
Yeah. I mean, I'd love to know the interface between bats and us.
Now we have a bunch of emails from the virology class at Indiana University of Pennsylvania.
Okay.
Remember, we didn't finish them. We have one from Heather.
Okay.
This is in reference to the discussion on junk DNA as a result of Stephen's email on TWiV 70. So she is responding to our answering one of the others. I tend to think that mobile genetic elements may not be junk even if they do not code for anything. Their purpose may simply be to accelerate mutations and therefore increase adaptive elasticity in a species. This could be of greatest benefit for rapidly reproducing organisms such as protozoa or insects. The organism that prevents these elements from infecting their genome might be unable to compete over time with one that has these elements. Perhaps once the elements can no longer jump, they would qualify as junk. I would love to hear your thoughts on this. Thanks for a great podcast.
My thought is that's brilliant.
It's a great idea.
Yeah.
Sure.
Yeah. There have been studies where people, this is coming out of a long time ago in my head, okay, but I seem to recall studies where people have tinkered with transposons to modulate their transposition frequency. And as you might expect, there's an optimum transposition frequency, optimum for both the host and the transposon. So this is a host-parasite relationship, basically. Or if they transpose too much, you wind up mutating the host to death and it kills it.
Right.
And a little bit of transposition, I think, is certainly good for the transposon, and maybe even good for the organism, for the host.
Yeah. So I'm from my textbook. It says that genetic analyses have documented a number of disease-causing genetic lesions resulting from transpositional events in humans. So they can insert into genes. That's mutagenesis. That could be conceived as an evolutionary driver, right? So it's the same idea.
Anything that increases genetic diversity is considered an evolutionary driver, yes?
Yes, absolutely. So that's why this thing's jumping around will mutate genes, as she suggests, and cause plasticity,
right?
Which is what you want. So yes, maybe when they stop jumping, when the transposase is dead, then they're junk DNA.
Do you think Barbara McClintock knew that?
Yes.
Probably. She was very smart.
Yeah, she was a smart lady.
Very, very smart.
Tyler writes, I am just curious as to what your opinions are as to whether or not the human Boca virus is responsible for respiratory infections. There is a common occurrence of human Boca in children who have respiratory tract infections, and I was wondering what your thoughts on or whether or not there's a correlation. And he sends an article about Boca viruses, which is called Correlation Between Boca Infection and Humoral Response and Co-infection with Other Viruses in Children with Acute Respiratory Infection. So these are pretty recently discovered polyomaviruses, right?
Parvoviruses.
Parvoviruses. Single-stranded DNA viruses.
Right. So the only other human pathogen in this family is the one that causes fifth disease, right? It's another skin rash. Okay, and everybody gets that. B19. It's another parvovirus. And you hear about parvoviruses, you hear about canine parvovirus and parvoviruses of other animals all the time.
Yeah. So these are all isolated from kids with severe respiratory disease, but they're often co-isolated with another virus.
Mm-hmm.
So it's not clear if they are the causative agent.
Or the helper.
Or they
just
exacerbate it.
Innocent bystander.
We don't know. My guess is that they are involved in disease.
That would be my guess too, yeah.
I think so. I think now I was looking through the literature, there are a lot of reports of isolations of these. And, the co-infection is not in every case. Sometimes these are by themselves. So,
right.
With disease still?
Yeah.
Okay, there you are.
So I think that they are. It's just that everyone's being very cautious about this.
Yeah.
And but
How long have we known about Boca virus?
Recently.
Thank you, Vince. How recent?
All right, let's Google it, Dick. When in doubt.
Have to ask these questions as a naivete
Alan,
person
can you look it up, Alan? Oh,
Alan's not here. Where is Alan?
Yeah,
he's not going to do it today.
I'm telling you, he's tapping those roots out there for the maple syrup. Now's the time.
2005.
That was the earliest I could find.
That's very recent.
First cloned in Sweden.
Yes.
Nasopharyngeal aspirates.
Interesting.
It's the fourth most common virus in respiratory samples, behind a respiratory syncytial adenovirus and rhinovirus. And do you know what Boca virus stands for, Dick?
I don't.
Bovine
Boca.
and canine. The two known hosts for other members of the genus.
The word Boca, and Rich, you should know this one, Boca Raton.
Right.
Anyway, that's good. Good question, Tyler. Christopher writes, thank you for the great podcast on reverse transcription. I'm wondering whether the retroviruses are more likely to integrate in certain areas of the chromosome, i.e., are they predisposed to incorporation into gene-rich regions or perhaps regions associated with the nuclear Cajal bodies?
Vince now feverishly is turning pages looking for Cajal.
No, I just taught a lecture on. The integration sites are, how should I say, random but selective. There are many sites in the host for integration. There's little sequence specificity, but the patterns are different according to the particular retroviruses. There is a preference for integration into DNA that are bent or unwound as a consequence of being wrapped around a nucleosome.
Oh.
So that's the one thing in common. So, how DNA is wrapped around the nucleosome, it's bent at a certain point, and those seem to be the preferred sites of integration. What's a nuclear Cajal body? Does anyone know?
I've never heard
I've just Googled it here.
How do you spell it?
Cajal.
Cajal.
No, spherical suborganelles found in the nucleus of proliferative cells. First reported by Santiago Ramon y Cajal in 1903, who called them nucleolar accessory bodies.
Using light microscopy, obviously.
Yeah. They have high levels of transcriptional activity. All right, so...
There are a lot of small RNAs in them, I think. A lot of these snRNAs and that kind of stuff.
Wow.
So I don't think that that's where these integration sites are. They're rather in bent DNA. Okay. Melissa writes, I had a question about prions. I got, by the way, guys, 2 cool articles for the next show on prions.
The Chinese study?
They mutate,
Oh.
Uh-oh.
What about the
that right?
fulfillment of Koch's postulates?
Oh, I didn't see that.
Oh,
I have to
yeah.
look at that. But I
was
found
a
an
good one.
article which shows that they mutate, they evolve.
Great.
All right, anyway. Sorry. It was stated on episode 67 that some prions were found to have beneficial attributes. I was wondering if the beneficial prions still produce the plaques that are characteristic of the infectious prions. Also, are all plaques produced by prions similar in structure? Are they easily identifiable as to what prion caused the plaques? So in yeast, there are genes with prion-like properties, which means they can template themselves to another protein. And they form these amyloid fibrils. That's the characteristic of the prion, templating their own structure and forming these fibrils. The plaque is what happens in a human who has the disease, or a cow or some animal with the prion
tangles,
disease.
they're called tangles.
Yes, so in the yeast you don't see the plaques because there's no CNS, obviously.
Right.
So it's not the plaque that defines the prion, it's the ability to template itself to another protein and form these amyloid fibrils. But in yeast, in fact, these prions are thought to be good, not cause disease, obviously. All right. Does that make sense?
Sure.
Yeah.
That's because yeast have very small brains.
Bigger than mine.
That's true.
Jessica, just a quick question relating to episode 67. Since HIV is mostly grown and active in the semen of infected males, where is HIV virus sustained and grown within the body of an infected female?
Well, I don't know that it's really accurate that it's grown and active in the semen of infected males, right?
It's
right.
It's got to be intracellular.
It hangs out in T cells.
That's
right.
Okay, and it's all over the place.
It's all over the place. It's in many fluids.
In both
right?
males and females.
Yeah.
Exactly.
And in fact, it's important to understand that it's transmitted heterosexually, both directions, from males to females and females to males, as well as homosexually, etc. So it's everywhere.
Yeah. It's produced mainly in lymphocytes, CD4 positive T lymphocytes, possibly some other cells as well.
Some
and
progenitor cells.
Progenitor cells, right?
That's right.
Right.
Bone marrow. And it is in many fluids, so semen is a good way to transmit it. But blood products work, but not respiratory secretions, because it's not produced in the lung.
That's right.
At least not in the lumen. Nicole writes, after listening to episode 66 about reverse transcription, wow, a couple of questions on reverse transcription. I had a question. If integrase and reverse transcriptase are the targets of an antiviral agent against HIV, is there any research using these enzymes against viruses related to cancer? They're different.
Yes,
they're different viruses,
the
though. They're different viruses.
Every drug has been tried against cancer, so you can just say yes to the question, but I don't know whether it was effective.
So the only other retrovirus involved in human cancer, there's HTLV1.
How about hepatitis B virus?
So Hep B, would have a reverse
uses a reverse transcriptase, not an integrase.
Right. So that's the other virus with a
good catch.
So I don't know if the drugs have been tried on that, but if I were doing the research, I would.
Sure. Yeah.
Well, let's see. Effect of zidovudine on hepatitis B virus replication. So zidovudine is AZT basically. So let's see. Does it inhibit? So they gave it to patients with HBV. After treatment, serum HBV remained positive in all patients and did not change. Our results show that AZT has no inhibitory effect on HBV replication. So that for AZT, it doesn't work. But the others, there are 20 other drugs. I don't know if they've all been tried, but Hep B would be the one. You're absolutely right.
But great question.
Yep.
Okay.
But cancer caused by HPV would not be touched because it doesn't have a reverse transcriptase or integrase. Yeah.
Likewise, what other cancer-causing viruses? Epstein-Barr virus? No.
Right.
That's not reverse transcriptase or integrase.
Hep C?
No.
Right.
So it's a candidate in a couple.
Okay.
That's a good idea.
All right. The last one's from Callan, who says, hello, I'm a student starting out my first course in virology. I listened to your recent episode of TWiV and thought it was informative and entertaining. I've heard about viruses mutating and infecting different species, like avian flu strains. I was wondering if the virus in most cases would continue to infect both the original species and the new species, or would it be limited only to the new species? Very good.
Good question.
Isn't it?
Very good question. Yeah. I would say you can all of the above.
Yeah, it goes both ways, right? Sometimes it goes in the new species and it stays there. Sometimes it goes back and forth. Yeah. So can we give some examples? So there are a number of viruses that are transmitted between humans and animals by mosquitoes. They go back, they replicate in both pretty well.
Sure.
What about like Ebola? Goes from a bat to a human. Does it ever go back? Or
I don't know. But how about influenza?
There wouldn't be much of a chance for it.
So influenza is the great one.
That's great.
In fact, last year's strain came from pigs to humans, and now pigs can be infected with that same strain again. Mm-hmm. Right. Mm-hmm. So it goes both ways, yeah.
Right.
So great question. I like that very much.
Mm-hmm.
Okay, some picks of the week. I'm going to start with Dickson Despommier.
Hey.
Who picked something called...
Well, it's an article about using whole genomic sequence data to identify a very, very rare disease. In this case, the disease is called Charcot-Marie tooth neuropathy.
Is this a dental disease?
No, it's not a dental disease. It's a neuropathy, but it's a demyelinating neuropathy. But Rich, I picked this one because of what you said last time, that you were going to have your whole genome sequenced. And the thing that jumped out from this news report of this, and there's another article too by
the Times,
the guys out in Washington, University of Washington, was that this guy paid $50,000 to have his entire genome sequenced. And you said you can only pay $1,500 to have yours sequenced. And
actually, they'll do it for free. Donations, but they'll do it for free.
Do they give you every base pair of your sequence, or
yes.
do they?
I don't know.
They do. You get 99%. Yeah.
Because this guy paid a fortune.
Well, he paid too much, but if he started 2 years ago, the price was higher. It comes down 10 times every year.
Comes back 10 times a year.
Absolutely
it comes down tenfold a year.
Amazing.
This guy, he paid $50,000. A medical geneticist. He's got a lot of money too.
Yeah.
No, I told one of my friends that I'd said, well, this personal genome project came up in the last TWiV, and Vince, you said you'd registered.
I signed up for it. Yeah.
Nice.
So I kind of hemmed and hawed about it. I thought it was interesting. I went home and told my wife about it. She says, "Oh yeah, let's do that."
Great.
And so we signed up, and we told our kids what we were doing. We'll leave it up to them. But as I said last time, I got twin daughters. I'd love to see how that came out.
Absolutely.
So anyway, so I sent an email to my friend saying that I'd done this, and he said, "You're wasting your time." And then he saw these articles too, and he said, "Well,
maybe you're
right.
not." So this guy
it's
had
better than a whole body CAT scan.
So he had Charcot-Marie tooth,
yeah.
And they identified a mutation in an obscure gene called SH3TC2, and that's it.
They found it.
It basically validates this kind of sequencing to identify disease bases.
Exactly.
And they say in this article, we are on our way to the $5,000 genome.
Exactly.
But we're already below that.
Yeah, $1,500.
Yeah. That's incredible, isn't it?
And this is a cool story.
I
So there's a
was.
Times article that goes with it, too.
Yes.
Which might be more straightforward for most people. And we can see this fellow here with a picture of his genome on a gel. There it is.
Right.
A couple of ethidium bromide fragments.
That's right.
Great. Thank you, Dickson.
You're welcome.
Rich, what have you got?
A book I read probably, 25 years ago.
You're not that old.
Oh, yeah. Invisible Frontiers. I read this book in 1986. How long ago was that?
He was an ingenue.
No, it's not 25. It's 15. It's 25 years. Invisible Frontiers: The Race to Synthesize a Human Gene by Stephen Hall. So Stephen Hall is a journalist, writes for a number of magazines, has written a couple of books, and this book is about the first efforts to clone and express a human gene. The obvious candidate was insulin because it has enormous therapeutic value and also because it's small, and they knew a lot about it. And there was basically a competition between, and it's been a long time and I can't find my old copy. Okay, so I was not able to refresh my memory. But the competition was between the Gilbert Labs at Harvard and Howard Goodman's lab at UCSF, and Art Riggs at City of Hope was involved. And I don't know if it was on his own or collaborating with one of the other labs. And a couple of different approaches were taken. Some were to reverse transcribe it and clone the cDNA. The Riggs group was synthesizing it from scratch. Okay, and this was all going on at the same time as the recombinant DNA debate was going on, because I don't know if many of the listeners will remember this or be familiar with it, but there's a very interesting era in science when scientists first developed the technology for cloning DNA, that is taking DNA fragments and being able to amplify them in bacteria and express proteins from them. They stopped and said, wait a minute, could this be potentially dangerous?
Right.
And had a conference to go over this, and had a self-imposed moratorium for a period of time, and it was quite a deal.
Asilomar.
A Silomar.
But the town of Cambridge in Massachusetts banned
it. That's right. That comes up in the book because there were experiments that these guys wanted to do.
That's right.
That were forbidden at Harvard.
Exactly.
They couldn't do them there. So all of this stuff is tied up in the book, and so it's
cool.
You picked a book last week that was a lab book, right?
Yeah.
And this is a similar type of thing where you get a real good insight into it. The science is very good. It's very well described, and all the personalities come through this, and the competitiveness, and et cetera. So it's
Natural Obsession was the story of the Weinberg lab.
Right.
These kinds of books are very interesting. I was in Cambridge about the time that you're at
MIT.
Yeah. And someday we'll have to tell the story, because the Italian mayor of Cambridge got involved. They had town meetings. It was very animated. When I got there, the Asilomar moratorium had just been raised, and I was able to clone polio RNA. Otherwise, I wouldn't have been able to do it.
Right.
So I remember David Baltimore saying, "Yeah, you can start this now. We just the moratorium is raised."
And I was thinking about this. I can't think of any example of recombinant DNA turning out to be dangerous.
No.
That's right.
Right?
No.
Yep.
So it really turns out to be okay.
Now, the scientists were just cautious.
Right. And that's good.
Yeah. But it turned out to be okay. Yeah. But I guess it's good that we showed some restraint for a while. Right. All right. That's really nice. I like that. I haven't read that one. I'll have to pick that up. I am going to read it on my iPad.
Well, that's exactly.
I'm not buying any more paper books.
Coming out April 2nd.
The store opened this morning at 8:30, and I pre-ordered at the moment.
Did you really?
The store opened, because all my books that I buy from now on are going to be on the iPad.
Oh, my goodness.
So if I go somewhere, I can take 5 books, and it'll weigh 1.5 pounds.
Save a tree. Read a book.
So I'll read it on the iPad.
How are you going to carry it, by the way? I looked up all their accessories, and I don't see any shoulder strap for the case.
It's only 1.5 pounds.
Yeah, but it doesn't matter. You still have to carry it.
It comes in a case.
Wouldn't you like this over your shoulder?
Dickson, it comes in a case.
I know it does.
That folds and makes a little angle so you can type on it.
But what if you're like in a subway?
Don't bring it in a subway. You could bring it to Florida because everyone's nice there.
That's right.
In fact, Rich, when I come to visit you in May, I'm going to bring my iPad.
All right. I can't wait to see it.
And maybe I'll even use it.
Okay.
My pick is a website which contains the proceedings of a recent conference that took place in San Francisco in February, the Conference on Retroviruses and Opportunistic Infections, CROI. It's an annual conference full of terrific retrovirologists, and all the talks, or most of them, were recorded, either video or audio. And so you can see all the latest research on HIV and XMRV as well. The retrovirus suspected to be involved
chronic fatigue syndrome.
Chronic fatigue and prostate cancer.
Yep.
So Steve Goff, our colleague, has a talk here. James Hoxie, Scott Hammer. So if you're these are all pretty short talks, straightforward. So I thought everyone would to hear about that.
Looks great.
It's really good stuff. I had another pick, but this came up, so we'll save the other one. All right. That will do it. All you new listeners, make sure you subscribe to TWiV in iTunes. Leave some comments so we can stay on the front page. We'll stay visible, and more people can find out about viruses. Because as you can hear, we have great conversations, pretty relaxed, enjoyable, and you can learn just by absorbing.
Fireside chat.
Fireside chat, virus talk.
That's right.
That's what this is.
That's right.
So we have a website, of course, TWiV.tv, where you can find all the show notes. You can send us your questions and comments to twiv@twiv.tv. We always love to get them, and we're almost caught up. We're doing pretty well. You can also find us at sciencepodcasters.org, promednetwork.com, and microbeworld.org, where you can find other science podcasts as well. Check out TWiP at microbeworld.org/TWiP, which is This Week in Parasitism. Dickson Despommier, thank you again.
Vincenzo.
Good to see you.
Racaniello.
No, say it right. Vincenzo.
Vince Racaniello.
Vinny.
Vinny.
Good to have
You
you back.
looking at me?
I don't have to thank Alan because he's not here.
That's right.
Rich, thank you.
You're welcome. I had a great time.
Always
so
fun.
did we.
Rich, we're not
So did we.
giving you too much work, are we?
No, I love it. Are you
All right.
kidding me?
Okay.
I learned so
I
much.
felt guilty this morning when I thought, geez, this guy's got to do a lot of work. I don't know if he really...
An hour of your time a day. Come on, you could be eating lunch and do this.
Thursday night is TWiV cram night.
Okay.
Yeah.
Okay.
All
I go
right.
home and I spend a couple hours cramming for TWiV.
See that?
I could spend another 6 or 8 hours doing it. I mean, 'cause there's a lot of material here, but I can't tell you how many times I've referred to things that I've learned since I started doing this show.
Yeah.
I told you a
Yeah.
while ago, Vince. I never have been a very good reader. I'm not good at keeping up with the literature, and this is marvelous motivation,
All right.
and it really broadens my horizons. It's wonderful. So don't give it a second thought.
So
I love it.
I'll forget about feeling
Right.
guilty. Okay.
No,
Good.
it's fine.
Because I tell you, I learn a lot too.
Yeah, it's great.
Oh,
did.
I
I was at a thesis defense yesterday, and I asked questions I would never have asked 2 years ago because I know much more. You know?
hope they gave you the right answers.
I look like a smart guy now, because I walk around and I know all this stuff.
That's right. Well, no one will ever confuse me for a virologist, no matter what. So I'm not worried about that either, and nor do I want to be confused as a virologist. But I think this is great.
And maybe when we're
really
done
do.
with TWiV, you'll be a virologist.
Because this integrates all of these thinking processes that you can now take, like Rich just said, into your own world and apply that to situations that have nothing to do with this.
All right. Sounds good. Sounds like a good way to end another TWiV. Thanks, everybody, for listening. We'll be back next week. Another TWiV is viral.